studied at the Pasteur and the Curie Institutes in Paris, receiving his PhD there. Was then a postdoctoral scientist at Mount Sinai with Miriam Mered, who many of you also know quite well. But then quickly, he was faculty there at Mount Sinai, but then quickly moved to A-Star in Singapore. And so we're very fortunate that in this sort of in this virtual platform, we can bring people together from all over. I don't know actually where Florent is right now, but I'm glad he's here with us. So we are very excited to hear his work. He has really done, I would say, very high impact science studying the development, the ontogeny of dendritic cells and other mononuclear phagocytes. And I think we'll have much to hear from him in the coming year. So I'm going to turn this over to you, Florent. You should be able to run your slides from there, I believe.
Thank you very much. So let me try to, if I click on share screen, it's telling me this will stop other screen sharing. Can I continue? Yes, okay. Let me know if you see my screen.
Yes, yeah, we see it. Perfect.
Okay, thank you very much. So thank you very much for having me. Good morning, good afternoon, good evening to all of you. Thank you, Paul, for your kind words, and really it's very nice to see so much of friends and colleagues, even through Zoom. So what I'm going to do, there is a little echo. So what I'm going to do today is to try to present you the latest development on our works and the works of others on DC heterogeneity and biology. And also the second part will be focused more on our work on LCH, in fact, using single cell technology in collaboration with Egle Kevadite from Karolinska, Matthew Collins also, and also supported by the Histiocyte Society. So, and all of this work have been mostly done in Singapore, but also in collaboration with a lab in Shanghai, Single Research in Singapore, as well as my new operation, my new lab that I've just opened in Gustave Roussy in Paris. So the lab focused on these two cell types, dendritic cells and macrophage, which are related cell type. Dendritic cells, as you may know, are sentinel of the immune system and antigen-presenting cells. Macrophage consider the scavenger of the immune system, they're the big eaters, and also sentinel of tissue homeostasis. Like macrophage exhibit specific functional adaptation to each tissue they reside and play crucial roles not only in immunity but also in development and homeostasis. But today, for the sake of the LCH topic, I will mostly focus on dendritic cell biology. And the aim of the lab is to better understand tissue immunity as well as tissue homeostasis and development through the better understanding of the biology of this two-cell lineage. And we have also like a particular interest in the ontogeny, the development of these cells to better understand their function. And also we like to group these cell dendritic cells and macrophage, which are monocytes, which are the precursors of macrophage, as a system which is called the mononuclear phagocyte system, the MPS. And through our work, but also the work of several other labs around the world, we have tried to propose a classification based not only on phenotype and function, but here based on ontogeny with the dendritic cell system, which is FLT3 dependent, with dedicated precursors in the bone marrow, with what we call the CDP for the common DC precursor, the precursor which is committed mostly to give rise to these conventional dendritic cells, the DC1 and DC2, and I will go back to this. On the other side, there is the macrophage, the CSF1 receptor dependent cells, which I've shown with Miriam Meral and others to have a very unique embryonic origin. And in the middle of them, it's a very puzzling lineage, the monocyte-derived cells, and that, of course, in steady state give rise to macrophage, but also in inflammation give rise to cells with features of both macrophage and DC, and often people call them inflammatory monocyte or monocyte-derived DC. And there is still a lot of work to be done to understand what does monocyte give rise to in steady state inflammation, and how should we classify this cell type. And I will come back to this in regard to LCH ontogeny, because it's an important aspect of it. I just wanted to show you this paper back in 2016 for people who are interested to understand how to identify and study this dendritic cells population. This work also that was led by Martin Guilliam to propose strategy to isolate and study dendritic cells in mouse, but also human tissue. So back to DC dendritic cells. Here you can see the first picture, in fact, taken by the late Ralph Steinman, that identified them in mouse spleen. And these cells are really in the middle, really controlling immunity against pathogen, but also maintaining tolerance against self-antigen. And really understanding how DC does and control this balance allow you to, of course, very relevant during this COVID time to maybe better understand new vaccination strategy, but also to better understand an autoimmune mechanism when the tolerance against self is broken. So some background: dendritic cells are residing in every tissue in your body. They are in steady state, in what we call the immature states. They are constantly patrolling, surveying their environment for like a challenge. And here, this immature DC will recognize some inflammatory signal, capture some antigen. What they need to do is to migrate to the lymph nodes in order to present this antigen to the T cells. It also will engage in the process of maturation. And then when they arrive in the lymphoid organ, will present the antigen capture in the periphery to T cells, also to B cells. And this will initiate immune response and will be able, in this case, to clear the infection, of course, in conjunction with innate cells from the innate system. And in fact, dendritic cells are really the innate control of adaptive immunity. And this graph, I think, captured really nicely and this was from Kanishi, Professor Kanishi in Japan, that shared with me his slide, capture really the essence of DC, able to recognize within minutes and hours of inflammatory response, of course, antigen, as I mentioned, but also the ability to understand through the unique expression of innate immune receptor the type of challenge that the system is facing, microbial products, danger signals that constitute now in textbook what we call signal 2. And this will lead to the activation, the maturation of DC that will lead to initiation of immune response when there is both signal. Absence of signal 2, signal 1 only lead rather to mechanism of tolerance and ignorance. Another important signal, which is signal 3, which is what are the cytokine that DC is secreting in order to polarize and initiate an adequate immune response. So this is general features of dendritic cells, but we know that DC can come in different flavor. And this graph, in fact, is already an oversimplification of the phenotypic heterogeneity of DC that was described until 2014, where this work led by Martin Guilliam and plenty of other teams have proposed to reunite dendritic cells in main flavors, in main lineage. And you can appreciate here that we know that there is what we call the PDC lineage, for plasmacytoid dendritic cell lineage, but we prefer to focus on what we call CTC, the conventional dendritic cells, DC1 and DC2, which have a unique ontogeny, a unique differentiation pathway, depending on, for example, for DC1 for BATF3 or IRF8, while DC2 rather depends on IRF4. And this very simple nomenclature, in fact, is reuniting all these different phenotype of cells described in the spleen or tissue, mouse or human. And this simplification have really allowed people to really, in fact, able to talk to each other across tissue, across species, to speak about the common DC lineage. And what is interesting is this dichotomy of DC is conserved. It's not only conserved at the ontogenic level, but also at the functional level. DC1 have been recognized to be quite relevant and focused, specialized in cross-presentation and Th1 response, so relevant for intracellular pathogen, while DC2 are more specialized in the induction of Th2, Th17, and making them more relevant to extracellular pathogen. If you think about a more evolutionary perspective where pathogen are really driving the evolution of the immune system. And PDC are very well recognized for their specific expression of interferon alpha and their specialized response, this involvement in antiviral response. So in fact, we we understand these two main lineage, in fact, as some kind of like anticipatory immunity, where these cells are specialized against a specific type of pathogen, anticipating the class of pathogen that will challenge the host. So why I'm spending time on this is not only to speak about function, but also to introduce the need, in fact, that the realization that the existence of two dedicated lineage of DC, the DC1 and DC2, that in fact at the time, 2014, were believed to arise from this DC precursor, the CDP, for the common DC precursor that give rise to what we call pre-DC, which is the most immediate DC precursors that migrate from the bone marrow to the blood to give rise either of this flavor of DC, the DC1, DC2. And at the time, it was not very well understood how this commitment was done, and also what was the sequence of events that could be at stake to differentiate into this different lineage. And at the same time, this is where my lab started to embrace new technology, such as single-cell technology, that are quite now really revolutionary approach and technology in immunology, but also, I think, in disease, and to better understand disease. So here's the pre-DC that people were thought were quite a homogeneous population, giving rise to these two types of DC. And this was a simple question that Dr. Andreas Schlitzer in the lab, postdoc in the lab, really asked, can we find specialized precursors or not? And are all DC precursors like the CDP or the pre-DC that give rise to DC1, DC2, are they all equal? And we will call them unprimed. Or maybe can we find already a transcriptomic pre-commitment in the DC progenitor pool at the level of the CDP and the pre-DC? And this is where the only way to approach this question when you don't know how to begin and how to identify these different committed precursors was to apply single-cell technology that will allow you to unbiasedly profile at the single-cell level the transcriptic profile of these cells, hoping to find some decommitment toward DC1 or DC2. And this is what we have done using a single cell where we have sorted a different population of cells, the CDP here and the pre-DC, using this C1 fluidine approach, where you can see here in the zoom, it is like a microfluidic chamber which have a little hole where the aim is to isolate in each of these little well one cell from which you will extract RNA in order to be able to access its single cell mRNA profile. This was really at the time low throughput approach, with each plate containing only 96 well plate. Now technology now allow you to sequence at the same time 10,000 of cells in each experiment. And what we have found is that this is a very interesting slide where we have identified a set of genes that we call DC signature genes versus macrophage signature genes, and we apply them to each of these step of DC differentiation. Pre-DC here is a DC precursor, so most of the cells, each dot here is a single cell, are expressing high level of this DC signature, so showing that in fact these cells are really committed to the DC lineage, and the CDP also, a significant amount also are committed to the DC because they have a positive score for these DC signatures. This was kind of like a positive control of the approach to show that those are DC precursors and they don't exhibit any monocyte macrophage signature. Now here on the right is the most important results from the paper, where we realized that when we looked now at DC2 versus DC1 signatures, so we identify genes which are specific of DC2 and genes which are specific of CD8, and we notice quickly that already not too much at the CDP level, but at the pre-DC level, which is the circulating DC precursor in the bone marrow, we could identify a significant commitment to either DC2 or DC1. Through like a complex bioinformatic approach, we look at genes that were specifically expressed in these cells versus those, and we finally identify a marker that could reveal, in fact, that pre-DC, the DC precursor were quite heterogeneous and could be subdivided in this sequence of events with this, what we call pre-DC2 that express Ly6C and the pre-DC1 that express also that we call double negative at the time, but that's also express a marker like CD24 or c-KIT, as shown by the group of Ken Murphy. So we refined further recently the sequence of events from this differentiation of DC, and we have identified, in fact, that PDC, I don't want to go into the detail, but in fact represent a lineage of DC on its own with its own precursors, and there is like a further sequence of events toward the differentiation, either to DC2 or DC1. So the main message here is like the commitment to become DC1 and DC2. In fact, in the DC lineage is taken before the exit from the bone marrow, before in the blood, and this is like an irreversible choice. So this is mouse data, how it works in human, and in human, in fact, what is fascinating is to realize that this organization is conserved, fully conserved, with the CLP giving rise to PDC, the CDP, the common DC precursor, that we have updated, mostly giving rise to pre-DC in the bone marrow that subcommits into early versus late pre-DC2, pre-DC1, that give rise to each of these DC1 versus DC2 lineage. And this stage of differentiation can be identifiable by using flow cytometry, and this is shown here. You can appreciate that all of these stages express different markers that you can map in different tissue in the blood, but also in disease, to try to look for maybe like cellular biomarker, DC cellular subset biomarker of maybe disease progression. And this also kind of approach of single cell have been also like used and applied by others. And here I want to highlight the work of Villani et al., also published at the same time with us in 2017. And the approach was way more ambitious than us. They really, like Chloe Villani, have this really like tour de force in terms of like high throughput sequencing of like really the blood, human blood DC repertoire, subset, cell population. And she came out with this like a atlas approach and annotated multiple different subsets. And she identified DC1, of course, DC2, but that came in maybe two flavors. And this is quite important. I will come back to this. She identified a DC4, a DC5, that is what we call the pre-DC and the PDC. She also identified different like flavor of monocytes. And of course, with time, people in the community have realized that there were like some slight like misannotation with this monocyte, this DC4, in fact, being a monocyte. There was some slight contamination in each of the approach, and also like this mono4 being a little contamination with NK cells, and this DC5 being our pre-DC. But I want to come back to, I think, one of the main discovery of this paper, which is very important, is the realization that this DC2, the CD1C DC, could come into two transcriptomate flavors. And what was really interesting is like in this paper also, like there is this arrow here suggesting that in fact what they call DC3 were very related, very close to monocytes. We have also, in collaboration with Joachim Schulze, proposed an updated map of this DC like subset heterogeneity, and this our work is posted on BioRevex. But the point here that I want to raise is, in fact, some DC subsets and some monocyte subsets here are still very close. And this is also, it might be seen as like details, but it's quite relevant for the work on LCH that I will show you after when you want to try to understand the LCH origin. So why are they so close? Is often because they are very close, sometimes very close in terms of phenotype. And we don't have yet, in fact, a clear way, a strategy to separate fully DC2 versus monocyte. And in contrast to the T cell population or the B cell population, which have clear lineage marker, CD3 for T cells or like B220, CD19 for B cells, here the problem is like the separation from what we call CD1C DC versus CD14 monocytes, as you can appreciate here, is not a clear population, but a continuum of spectrum of cells. And the problem of like flow cytometry based approach or sorting based approach is like you need at some point to put a gate to separate the cells. So you could imagine that some of these cells, CD1C cells, could still be DC, but very close to the monocyte gate, and some monocytes also could contaminate DC, really showing that these CD1C versus CD14 markers are not resolutive enough. So the aim of the work of Charles-Antoine Duterte was to try to find a better combination of marker to be able to better delineate DC versus monocytes in human tissue especially, and try to understand, clarify what is the nature of these intermediate cells. So this, I don't have so much time to go into this pipeline, but this was with the help of Etienne Betch and Evan Newell, and they recently published an updated approach, was the ability that they designed a way to screen 332 markers using flow cytometry on DC and monocyte. And we came with like a very predicted map of marker expression for these 332 predictor marker using bioinformatic analysis. We refine on each of the population the best marker that were highly expressed on each of these different populations to come with like 92 discriminant markers that allow a clear separation versus DC2. Here you can see in orange versus monocyte. And then we asked the bioinformatician to tell us what are the best markers that are able to separate monocyte versus DC. And we came with CD14 for monocytes. Of course, we came back to some marker. Those people were not wrong, but we also added CD88 and CD89 that are also able to better separate monocytes here versus DC2. And also we came with also like FC epsilon alpha and HLDQ as best marker for the CD1C DC2. So this sounds like, again, trivial and a game of adding more complexity, but when we added these markers to separate monocyte versus DC, we realized that a significant fraction of the DC2 here, gating only on DC2 using this new gating strategy, express CD14, implying that most of the time, or often, using only CD14 to gate on monocyte imply that a lot of this monocyte strategy, isolation, purification, were heavily contaminated with DC2. So arguing for maybe to revisit, like some aspect of antigen presentation that have been attributed to monocyte. We look at the cells by electron microscopy, what we call this different subpopulation, especially the CD14 here that will have fallen in the monocyte gate, express CD163, but look like dendritic cells. What was the relevance? When we looked at the transcriptoral profile of these cells, we quickly realized that they were inflammatory cells. They were really exhibiting a very high level of inflammatory signatures, especially a pathway related to systemic lupus erythematosus signaling. And when we looked after, for example, now in lupus patients, in systemic lupus erythematosus SLE patients, we noted that the population of DC3, the CD163 positive, were quite enriched, as you can appreciate here. And this is like the quantification. And also what was striking is the level of the percentage of this DC3 population that before was in the monocyte population, but we could extract by the gating strategy, was correlated with the disease score. So maybe implying that this population of cells discovered by single cell and also like by a high dimensional profiling of markers is probably playing an important role in the pathophysiology of lupus. And we are now extending its characterization and in other diseases. I just want to before to move in the LC-Watch work, to just warn you about the nomenclature clarification. Recently, a lot of single cell paper have reported a lot of different population of DC, and often have annotated them in a numerical way, DC1, DC2, DC3. And there is several papers which have reported DC3, which are not at all related to the cells I've just discussed with you, but related to this, what we call MREG DC, which is the migratory or the regulatory DC, dendritic cell regulatory program, described by Miriam Merad recently. So just beware, the nomenclature is still a little bit tricky right now, and we hope to clarify it very soon. So now with all of this basic of dendritic cell heterogeneity and biology and this different population in health and disease, let's move to Langerhans cell histiocytosis And I want to present some work which have been led by Dr. Egle Cavadete from the Karolinska, and again supported by the Histiocyte Society. So what Egle wanted to do is to apply a single-cell analysis of LCH cells to identify their core signatures and heterogeneity, and to validate markers and pathway on protein level. So the study design, is we have collected 4 LCH lesions from pediatric patients. We have done single-cell indexing as well as single-cell RNA-seq. So we could, on each cells, have protein expression, but also RNA profiling to assess and evaluate lesional cell identity and LCH cell identity. You then move to look at interpatient and inter-experimental approach. We validated using protein level, but also we integrated our data with published data set, and I will come back to this. And we validate this patient and tissue heterogeneity and assess also the spatial distribution of these Langerhans cells. So this is the compilation of the 4 patients that we have analyzed. This is the 4 patients here with the distribution. And in these 4 patients, we have identified, of course, several cell types, including monocyte, DC1, DC2, pre-DC, PDC, but also, of course, LCH cells that are importantly clustering very, very, very uniquely and are not close to monocyte and not close to normal DC1, DC2, pre-DC, or PDC. And Langerhans cells, as expected, express high level of CD1A, CD207, and do not express several other markers which are specific, for example, of monocyte. And this is the annotation. This is the gene which are specific of each of these clusters. And here, for example, again, and the clusters, these are the genes which are expressed by the other cell cluster. And importantly, among these 4 patients, there were not too much specific patients, it was quite equal in each patient. What is important is to recognize also that, again, it's important to be able to delineate the LCH cells from other DC population and to consider that maybe some DC population have been mistakenly integrated in some LCH data set. The core signatures of these LCH cells are 207, CD1A, MMP9. While with other markers, we can isolate here the CCR7, LAMP3, which are the MREG DC that could have been mistakenly taken for LCH, and also here the CLEC9A, the DC1, and other different population. And again, the identification of this MREG DC here published by Miriam Merad as a tiny subset here. You can appreciate here the MREG DC are here with unique program, very different from LCH cells. So what about LCH? When we looked only at LCH versus the rest of the myeloid cells, including monocyte and DC, and we look at genes uniquely expressed or highly expressed on Langerhans cells, of course, we noted CD1A, CD27, the classical marker. But when you look at pathway analysis, we noted that there were really upregulation of pathway implicated in mTOR signaling and also related EEF signaling that, in fact, are related to senescence. In fact, the LCH cells look really not low proliferative, metabolically impaired senescent cells. And this is the expression of key senescence-related genes, such as CDKN2A, LMNA, CKLF, PAC1, CCND2, FCGP, PTNF, which are highly expressed, as you can appreciate it, in the LCH cells. And we were very happy to see that this was purely aligned with the recent paper from Miriam Merad that have showed that really the BRAF mutation, which have been implicated in Langerhans cells histopathology, really induced senescence in Langerhans cells histiocytosis pathophysiology. So this was in line with Miriam's finding. And we wanted to go further in the characterization of LCH cells. And we have tried to now isolate only the Langerhans cells and try to see how heterogeneous they were. And using a bioinformatic approach, it revealed that LCH cells among these 4 patients could be distributed in 2 subpopulations. It's here the cluster 0 and the cluster 1. And when the cluster 1 here, you can see the pathway like implicated like mitochondrial dysfunction, oxidative phosphorylation, suggesting these cells are like in situation of stress, while the other one have like other marker, including HLA-DP, as well as like other like unique marker with this pathway, maybe indicating here like some EIF2 signaling and other lipid representation by CD1A. So two different programs among lung LCH. So far, what I've shown is like LCHs can be delineated from the normal counterpart using single-cell RNA-seq. Their specific signature point to senescence, and there is clearly two cluster LCH cells. So the next question is like, what is the developmental relationship between this cluster and the normal counterpart? And this is where the previous work I show you between DC2 and DC3 monocyte need to be taken into account, and this is what I want to show you here. The DC heterogeneity and the mononuclear heterogeneity with monocytes that come in different flavor, but also DC1, DC2, as well as DC3. So, what is the precursors of LCHs? Do they come from DC1, DC2, DC3, or monocyte? And also, I want to point to the beautiful work of Carl Allen, Miriam Merad, and others, Matthew Collins, which have shown really like that among this like myeloid cell differentiation, so you know that there is like a clear myeloid ontogenic path for LCH, with maybe the acquisition of the BRAF mutation at different level that could play a very important role in the distinction between high risk versus low risk LCH. Of course, the earlier you get the mutation, the higher the LCH is severe, while the later you get the mutation, the less it's severe. But now the key question is like, if you acquire this BRAF 600 mutation, whether it also can be acquired maybe at different stage, but also in different cell types at lineage, DC versus monocyte. So what we have done exactly as the same type of approach that I showed you before on trying to understand the transcriptomic commitment of pre-DC to DC1, DC2. What we have done is we have designed a monocyte DC3 signatures, because those cells are very related and very difficult to tease apart, and the DC2 signatures. And we have applied these specific gene signatures towards our two clusters of cells, the cluster cells LCH0 and the cluster LCH1. And we can see that there is some kind of trend with LCH0, the cluster 0, having like a clear more DC2 signature versus the cluster 1 having a more monocyte DC3 signatures. And when we projected this, for example, here, when projected this population, uniquely this like more DC3 monocyte versus more DC2, you can see that they cluster at different area of the 2LCH cluster, suggesting that this cluster here is mostly related to monocyte DC3, while this cluster here is monocyte. It's more related to DC2. So it suggests just that maybe in the LCH cells, you could have two types of LCH. One is more related to DC2, and one which is more related to monocyte DC3. So the premise, our approach, we were a bit limited by cell numbers, so we wanted to extend this approach to like other data sets. And luckily, there were like this beautiful work from Arbrita and al. That have like really released really like a beautiful 10x chromium data set with 7 patients. And what we have done is we have integrated our SmartSeq data to their data, and we have looked at how do they overlay. So extracting all the Arbrita data, we could find 3 clusters, cluster 0 here in red, 2 and 1. And after when we overlay on top of them our cluster 0 versus 1, we could see that cluster 0 LCHs were more like accumulating in this area, while our cluster 1, which were related to monocyte DC3, were more accumulating here. So suggesting that also in a different data set in other patients, this dichotomy also could be seen. And we have done exactly the same approach, where we have applied monocyte DC3 signatures towards this 3 major subsets, mostly contributed by the Arbrita dataset. And we can see also a trend with this cluster here being more related to DC2 and the other one more related to monocyte DC3, suggesting maybe like a dual origin of Langerhans cells. And when we applied, for example, like algorithm that predicts developmental trajectory in single cell using the stochastic or dynamic model, we could see this would reveal, in fact, the convergence of two pathways, this cluster that go toward the center, while also the blue pathway was moving this way. So maybe those LCH cells are not very distinguishable, but they could arise from both DC2 or monocyte DC3. And of course, the key question are both population neoplastic, or do they carry the BRAF mutation? And in our data set, we couldn't see any difference between these three clusters. And there are equal amounts of the BRAF mutation, suggesting that this was not a main confounding factor. So far, LCHs show polarization toward DC and monocytes. We predict two major developmental trajectory, maybe one arising from DC2 versus another one arising for DC3 monocyte. And both LCH clusters carry the BRAF mutation, but not the healthy counterparts. And the last slide I wanted to show you is like, can these two LCH populations can be found, especially in tissue environments? And this is why we wanted to validate the presence of this subset in tissue microenvironments, where we have acquired a different LCH lesion and control tissue, bone, skin, lymph node, bone marrow. We have like tried to image outside versus inside of the 207 lesion through like a new approach, which is called Maxima, and apply some segmentation algorithm. And the data were quite exciting because among here, like the 207 lesion, you can see the 207 cells that LCH cells, which are contained here in this zone. We have been able to image some class 2 higher cells versus also some CD44 positive cells, and we could we postulate that the MHC class 2 high cells correspond to the LCH0 cluster, while the CD44 correspond to the LC1 cluster. So this still need to have further validation, but it suggests that maybe these two flavors of LCH can be found in LCH lesion. So in summary, LCH may arise from both DC and monocyte DC3 lineage. We are trying to validate this approach using protein analysis, cross-dataset integration, microscopy, and interpatient comparison. And also, questions like how can this knowledge now not be used to design target approach for complete elimination of LCH cells? And we're still trying to better model the LCH pathophysiologic acquisition program. I want to highlight also like a paper from the group of Bing Liu in China, where I and Egle also have contributed, where also like they provide a really heavy transcriptomic landscape of LCH cells and really unveils like this universal MAP kinase activation in circulating cells, not even LCHs, but already non-LCHs in the blood, and potential therapeutic mechanism of BRAF inhibitor in pediatric LCH. They provided data of kids treated with BRAF inhibitors, and a decrease, and they find a biomarker, which is decreased frequency of circulating PDC that was significantly associated with disease severity. And on this, I'm going to stop here and thanks the people which have done the work, which this is like the first part on the DC part, my lab mostly in Singapore, but also in China, and key collaborators. And also, of course, in the second part, focusing on the LCH work, Dr. Egle Cavalidator from the Janninger group, before in Karolinska, all of our collaborators, Matthew Collin in Newcastle, and also all the doctors and nurses and patients, and also the support from Histiocyte Society, and also like seeing Paul today, and all the Nicolas Symposium like friends and members. And thank you very much for your attention.
All right, so I'll give you some actual applause.
Thank you. I hope. Yeah. Thanks.
It's a deafening crowd if everybody could do it. But we have a few minutes before we move on to our next session, which is updates on registries. But we have a few minutes for questions right now. So if you have a question, just use the raise hand feature, and we can unmute you so you can ask your own question. But while people are doing that, I had a very simple question for you, Florent. A lot of times monocytes are defined by not just CD14, but CD16 and 14. And I didn't really hear, where CD16 falls in all this ontogeny, because it seems to me there's some confusion about kind of these 14, 16 double positives. Could you comment on that?
So the main population of monocyte, of course, are the CD14s, and there is after this two-site population, one that is double positive for 14, 16, which are the non-classical monocyte, but they represent a very tiny amount of cells. And there is after the last subset, which is the CD16, which is really, which is 14 low. I think there is evidence, the work of Simon Yona, at least in human, using labeling, tracing, heavy water tracing in human, in healthy patients, suggests that this CD14 give rise to this CD16 in a precursor relationship. So they both 16 and double positive seems to arise from the 14 monocytes, which are the major population.
Mm-hmm, mm-hmm. Got it. Okay. And then now let's see. I don't see any hands raised at the moment.
I see Astrid. There is Astrid.
Astrid?
Yeah.
I raised my hand to Mike.
Okay. Oh, there it is. Sorry. Here we go. But let's let Astrid. Why don't you
unmute yourself?
Yeah, you can unmute yourself.
Yeah.
Okay.
Please.
Well, thank you very much, Florent, for this beautiful talk and nice overview of all your very interesting data. I have a very simple question for you. I'm very intrigued by the fact that you find LCH cells in tissues expressing CD44. CD44 is quite a conventional homing receptor. So what do you think is the biological function of this CD44 expressed in the tissue?
I think these cells could represent cells that just arrive and cells that just have been recruited in the environments, and could be represent some kind of like a early stage LCH cells, which after will maybe lose CD44 and acquire class 2. This will be like the most simple but very hypothetic response.
So you don't believe that the cells actually upregulate the CD1A and or the CD207 in the tissue?
Oh, they will. But I think you might have a stage where cells, you could have cells that have the BRAF mutation, which are not yet fully LCH-like cells. For example, in the paper from the group of Bing Liu, we clearly show that cells which are non-LCH cells already in patient exhibit huge activation of MAP kinase already in the blood. So there is maybe a priming, and then once the cells arrive in the tissue, they could, of course, acquire the CD127. I think this is a clearly tissue-specific TGF-beta notch-dependent mechanism, but you could have a stage that could be CD44, but this is really hypothesis.
Okay, so because I was expecting another kind of chemokine receptor to go to the skin, not necessarily CD44, but maybe those particular chemokine receptors have already been downregulated upon the entry in the tissue.
Yeah. But we didn't stain everything, so we cannot. But if you have some idea, I would be happy to collaborate and just test a few of your ideas, no problem.
Okay, thank you. All right, Ashish, and then we have Daniel after you with another question. Yeah.
Again, I echo what Astrid said. Fascinating. So if you define these two populations, presuming that they exist in every LCH patient, what you call 0 and 1, and one of them has a more DC-like features, the other one has more monocyte-like features. Can you speculate, are these fixed, or can they actually fluctuate between the two populations? How dynamic or how fixed is this phenomenon?
So again, this is purely speculative, but what is great and bad with all the single cell is like you can speculate a lot, but the danger is to speculate too much. But let's speculate a lot first. So what for me intrigued me is the result from this like velocity where you can see these two subsets that converge. And I think the fact that you see them converging and then in the middle you start to lose this separation between DC2 and DC3 monocytes. So what I interpreted is like cells arrive, I can see DC2-like arrive. There is also monocyte DC3. They enter into this kind of tissue program and they become full LCH cells. And there is the loss of this DC2 versus DC3 identity that get overwhelmed and reprogrammed. Basically, the CD1A207 program masked then after your cell of origin. What is quite intriguing is that the DC2 counterparts proliferate a bit. while the monocyte DC3 do not proliferate. So there is two ways to interpret this. Either the DC2 is more like a precursor, but also one of the biggest difference between DC and monocyte is the proliferation capacity. So this is still an open question.
Thank you.
All right. Okay. And then Daniel, you had your hand raised as well.
Oh, thank you so much. Great talk. This could be a little bit out of LCH context, but then again, cross-speciality contamination and knowledge is so important. I have not been aware of the importance of FLT3 in the development of PDCs. Do you have any comments on the potential role of FLT3 inhibitors in blastic, plasmacytoid, dendritic cell neoplasms? Which is a rare disease, but then again, sometimes we give lymphoid-directed, sometimes we give myeloid-directed therapies. And if you're interested, we could have some samples for you in a freezer.
Of course, I will be always happy to discuss and collaborate. So yes, it's a good question because it's a very confusing field. I think clearly DC are FLT3 dependent cells, the conventional, but the FLT3 dependence arise quite late. Maybe at the CDP level and the more committed. While the PDC, if we believe that they arise mostly from lymphoid precursor, I think their FLT3 dependency maybe come mostly from their CLP, kind of like CLP2 or lymphoid. B cells are FLT3 dependent. Some ILC are FLT3 dependent. So it might also reflect a different path, but those are really open questions, very debated. But happy to discuss for the PDCM.
Great. All right. Okay. And then there was also a question in the text from D. Samuel asking about what gives the LCH the characteristics of senescence. But maybe we should refer him to the paper, right?
Yes, but probably a combination of tissue residency plus the MAP kinase activation, metabolic disruption, and this cell doesn't want to die. This is why they are senescent, they accumulate. Okay, thanks. Sorry for being late.
That's okay. We're actually just on time. Florent, thank you very much.
Thank you very much for
that excellent talk.